So we have finished chemistry. And I know I say finished chemistry. What I really mean is we have gotten through the absolute basics of chemistry, and we will revisit it again and again and again and again. We're now entering into the cell. The cell is probably what you guys have experienced previously in biology, learning the parts of the cell. You may have made a model of the cell out of food. Do you guys remember doing that, making a model of the cell out of food? I would appreciate that, because this is the point of the day where we're starving. Jello mold or some such. But you guys have been learning about this. My daughter's learning about this now in 7th grade. She's learning all the different parts of the cell and what they do. My son's figured out that a cell exists. That's about it. So today we're going to go over a big overview of the cell. We're going to talk about, at a macro level, what goes on, because I want you guys to have a big picture image. After today, we're going to go nitty-gritty into each little piece of it and how they relate to each other. But for today, it's to map it out, see what the point of each of the sections are, and understand how everything connects. It's all about connections. I can't say that enough. The cell is a factory for producing you. All of the components. So, to see the cell, we need to be able to use this powerful new tool of science, the microscope. Now, it's new because it's been only around for about, 300 years. Considering human history, fire's been around a lot longer. But the microscope's super important. It was originally invented by a gentleman named Robert Hooke. Now, there should be an umlaut, I believe, over the, oh no, there's no umlaut in Hooke, sorry. So it's Robert Hooke, and what he did was, you guys, have you ever played with somebody else's glasses or your own glasses? You take glasses and you go like this, and you move them back and forth to your face, and it looks like the things in the distance are getting bigger or smaller. Well, what Hooke did was he noticed that effect. If you take a lens and put it forward, it'll magnify something or make it smaller. But then he did the next obvious step, which is he took two lenses. He put one here, he put one farther out. And he looked at that and said, oh, look, now I can see things that are far away and they're very close. Or, sort of like those magnifying glasses. You know what I'm talking about, magnifying glasses? Think pirates? Yeah, good. Magnifying glasses. Not magnifying glasses, spy glasses. Magnifying glasses what Dr. Fox uses to read his mail. So they had these spy glass like that. Now they've had that for thousands of years, but what Hooke did was he took it from looking out into the distance, and he did the next obvious thing, which is to look down. Because, we all want to see what's really up close in front of us. He looked at the first thing he had available, which happened to be a wine cork, so that says a lot about his personality. He looked at this cork, and in this cork, what he noticed was that it was made of, do you guys know what cork is made of, by the way? Cork is wood. So he looked at this cork, and it's dead wood, bark. And he noticed that it was a whole bunch of squares, sort of like that. Bunch of rectangles lined up next to each other. And he said, those look like the cells of monasteries where monks live. So he started referring to these as cells, and the term has stuck ever since. About 1775, so 100 years after Hooke, a gentleman by the name of Anton von Leeuwenhoek. Now there's the umlaut. Although I couldn't figure out how to put the symbol in PowerPoint, so sorry about that. So Anton von Leeuwenhoek, he refined the technique. Instead of just taking two lenses and putting them on top of each other, what he noticed was that these lenses did not have good glass. Have you guys ever looked through the bottom of a glass bottle? Hypothetically, some of you guys might have finished drinking something, let's say something, and you finish it up, and then after you finish drinking this substance, you look through the bottom, because that's how good of a mood you're feeling after you've gotten through it. And what do you notice when you look through the bottom? It's all distorted. If you didn't, someday in the future, you will look through the bottom of a bottle and think to yourself, wow, that looks really weird. You'll probably think back on this class and be like, that guy told me that. I wonder what his name was. You look through the bottom of the bottle, it's all distorted. That's because the glass is not very high quality glass. It has a lot of bubbles in it. Imperfections. Leeuwenhoek, he got rid of those imperfections. He made it so you could see crystal clear through these lenses. That increased the sharpness. It made it so things were no longer blurry. Now, you didn't just get to see little square rectangle things that were out of focus. Now you could see living, squirming things that were in focus. Cool thing about Leeuwenhoek, he wrote an article about it, but he was not a scientist. He was a salesman. And you know what he sold? Glass lenses. That's right. He wrote a paper as advertisement for his actual job. People started getting weirded out, though, because if you look at a living person's skin, you have what's called a microbiome living on it. You are covered in living organisms, feeding off of your dead skin material. Some people have like mites living in their eyelashes. These things are everywhere, and you could suddenly see them with the scope, and you couldn't see them with your normal eyes. It's really funny. If you're in a relationship with somebody, or if you're not and they're just really creepy, whatever, you're staring deeply into their eyes, look up at their eyelashes, because there might be something crawling on there, a little eyelash mite. It's living off of the sebaceous secretions from their eyes. It's there. It's not cool. I know you guys will. Now you're going to go and check. You're like, and you're also thinking, man, that's good. Do I have those? How would I know? So you'll find yourself at home in the mirror, staring at your eyelashes. It happens. Welcome to biology, where you start to question everything. You've got, you know your armpits, right? Well, you don't know them. I guess you might know them. You can't really look at them too well. They smell bad. I think we can all agree on that. That's why we use deodorant. If you don't use deodorant, it's even worse. In your armpits, you're going to have these organisms, these bacteria growing that emit these smells. How do you get your armpits to not smell anymore? Not trying to trick you. And it's not use deodorant. You wash them, that's right. You know there's several people out there who are like, oh, I stink, just more deodorant. That doesn't get to the root of the problem. The problem is you've got these organisms living on your body. You couldn't see those before Leeuwenhoek. Suddenly now, we can see these things. And they're moving, and they're alive, and they're on you, and they're eating pieces of you. And we're okay with that. Heck, anybody ever had, you don't raise your hand. If you had athlete's foot at any point in your life, it's going to cause little bits of skin to flake off the bottom of your foot. It's itchy. What's happening? You've got fungus growing all through underneath your dead skin cells, devouring dead skin cells. Living organisms growing in you. And we couldn't see them until now. So that's the history of the basics behind how we got our scopes. This leads into this idea of what's known as the cell theory. A gentleman by the name of Matthias Schleiden, German gentleman. Schleiden studied plant physiology. He studied how plants work. And he noticed that in all plants, they had these cells, and in all the cells, there's this dark spot. He had no idea what the dark spot is. From your labs, what do you know that dark spot was? The nucleus. He stained it with iodine, just like you did. So he noticed this, and he said, this is interesting. And he looked at plant after plant after plant, and every single type of plant he looked at under the scope, he found this nucleus. So he told his compatriot, Theodor Schwann. Theodor Schwann was an animal physiologist, and so he studied animal cells. And after Schleiden told Schwann, and I assume it was over bratwurst and beer because they're German, and that's the way the stereotype works. After bratwurst and beer, they talked, and Schleiden told Schwann, hey, I've seen these, in his German accent, I've seen these dark spots in the cells. I think they're in all plant cells. I think they might be in all cells. Well, Schwann said, I think you may be right. I've seen those in animal cells. I'll write a paper about it. And then Schleiden said, no, I'll write a paper about it. And then they raced to publish. Their friendship, by the way, got ruined. They raced to publish. Schleiden published first, followed by Schwann the next year. And that sort of indicated that all living organisms are made up of cells. 20 years after that, you have a Russian gentleman by the name of Rudolf Virchow. Virchow proposed that all cells come from other cells. He got that by looking at cancer cells. He noticed that all cancer cells produce more cancer cells. They don't make normal cells. Normal cells make normal cells. Cancer cells make cancer cells. So from that, he proposed this idea that all cells come from other cells. Now, between these three gentlemen, we get what we call our unified cell theory. If you haven't started writing things down, this is the point. Unified cell theory. All living organisms are made up of one or more cells. Cells are the smallest unit of life, and new cells come from pre-existing cells. This holds true for Bio 101. When we get to Bio 102, we're going to start questioning some of these. All living things are composed of one or more cells. Is a virus alive? Is a prion alive? Those are things we'll talk about in a later course. But for you guys at this level, this is the basic definition of the cell theory. Everything is made up of cells. Cells are the smallest unit of life. And all cells come from pre-existing cells. Of course, that also leads to the chicken or the egg problem. If all cells come from other cells, which was the first cell? Again, Bio 102 we cover that, not Bio 101. Just go with me for now. We're able to look at these cells using microscopes. And we have three kinds of microscopes, of which we will use only one type in this course. The light microscope. I mean, check it out. That looks very, very similar to what we have in the lab. It works in the same way. All light microscopes work the same way. They've worked the same way since Hooke's time. The idea is you've got two lenses positioned on top of each other. In this case, there's a mirror reflecting downwards that causes the image to be transposed. And underneath, there's a light source of some kind to shine up through the object. This only really works on objects that are translucent. They let light through. And you can get some pretty good images. This is Elodea. It's a plant cell. This is what you would get out of a light microscope. Nice colors, but not much magnification. You can see the individual components, but you can't make out what's going on inside of them. The other two classes of microscope are going to be the transmission electron microscope and the scanning electron microscope. These are much more expensive machines, ranging in the close to millions of dollars each. I think the closest one to us is there's one in Virginia Tech, and there's one out at Duke. The transmission electron microscope is going to send electrons bombarding through a sample, and it'll collect the electrons that made it through and plot them out on a film on the back. Now, to create a slide like this takes about 3 or 4 months. You have to go through a big process called microtoming, and you have to do everything in a complete sterile vacuum. Very difficult project. Odds are you will never have to work one of these until you're in graduate school. And even then, they're going to be leery about letting you use it. A scanning electron microscope scans along the edge, uses electrons, but scans along the edge of a cell. Sort of like if you've been to the grocery store and you've seen a laser popping up through, when you're scanning your groceries. Good. So it will follow the contour of that thing. If you were to put your hand in front of that scanner, it will follow all the contours of your hand, sort of running along the edges of it. That's what's happening with this the scanning image. It scans along the outside of the cell. That gives you a really good idea of what's going on the outside of the cell. Three-dimensional image, it looks awesome, but it's only of the outside of the cell. You can't see what's going on the inside. To know what's going on the inside, you have to use a transmission electron scope. But like I said, you guys won't ever really have to use one of those. Some of them are, 15 feet tall? Pretty tricky. So don't really write this stuff down on this slide. This slide is how I set up the course. We got 4 big components. Matter. Matter's everything we've talked up to right now. That's all the stuff about chemistry, all the stuff about physics. Check mark on that. Next up, we've got energy and organization and information. Today, we're talking about the organizational aspect. Everything in the cell has its proper place. Everything in the cell, there's a reason for everything to be in the place that it's in. After we talk about organization, we're going to talk about how the cell gets its energy, how it transfers it from one form to another. And then we're going to talk about how information is exchanged in the cell, how you can move information from one generation to the next. That's genetics. So for this part of the course, we're going to talk about organization, and then we're going to talk about energy needs of the cell, followed by how information is passed from one generation to the next. We can classify cells in one of two ways. They're either prokaryotic or they're eukaryotic. Prokaryotic cells are like bacteria and archaeans. They have very, very, very little compartmentalization. Very simple structures. That's compared to eukaryotic cells. Eukaryotic cells have more complex structure. They are compartmentalized. So prokaryotic are not compartmentalized, eukaryotic are compartmentalized. As far as compartmentalization goes, so something that's not compartmentalized, your room. Think about that. If you don't have this very controlling personality, think about, or a room of somebody you know, they might have everything splayed out everywhere. Laundry. Let's talk about laundry. All right, with laundry, you have a basket, and into this basket go dirty clothes. Does that make sense? Do you guys have this at your house? I mean, some people may not. There might just be a spot, a pile on the floor. Let's say you just have a random spot that you normally throw your dirty clothes. Does that work for you? Okay. That would be sort of non-compartmentalized if there's just a spot. There's no special compartment for it. There have been times in your life where you've done laundry and you didn't fold it up and put it away, right? And you have then piles form. You have your clean clothes and your dirty clothes. And occasionally, because you're in college, and I was there at some point too, you have your middle pile of, well, they don't smell bad yet, and I could probably still wear it again. This is getting a little too close to home for some of you guys. These could be all over your room. Compartmentalization would be, let's put everything away. The socks go in the sock drawer, and the underwear goes in the underwear drawer, and the, pants go in the pant drawer, the shirts go in the shirt drawer, whatever. That's compartmentalization, putting things away in their proper place. Non-compartmentalized is, you've got an idea of where all the clean clothes are. They're in that pile. It doesn't have a compartment it goes in, but they're there. You can still get to them. Children are great at non-compartmentalization. Some of you guys may have kids someday. Some of you guys were kids. Okay, most of you guys were kids. All of you were kids at some point. When you were a child, what did you do with your toy? You had different types of toys. You played with them. Did you put them away after you played with them? No, you didn't. But you still knew where they all were. You could have, let's say, I don't know, Transformers, Legos, and another toy. What did you guys play with when you were little? Welcome to community college. All right, so you've got dirt, rocks, and sticks. Hypothetically, dirt, rocks, and sticks. Different things. And you might play with your sticks one day. You might build a little tent out of them, or a shelter because you need one. So you built this shelter out of sticks. This is turning into the Three Little Pigs. You built the shelter out of sticks, and then you abandon it, and you're like, I'm going to play with my dirt now. I'm going to turn it into bricks. You still knew where those sticks were. Your parents were like, clean up your sticks. You didn't need to. They were fine where they were. You're good with this. Even though you did not compartmentalize where all of your toys were or your lawn objects, you knew where everything was and could use it at any given point. Does that make sense? That's non-compartmentalization. Compartmentalization is everything has its proper place. I will take my sticks and I will neatly arrange them right here, and all of them will stay right here because that is the stick area. Over here, I'll put my rocks. Over here, I'll put my dirt. And life is good, for they are all separate. You know people like this who have desks. that are perfectly clean and everything is exactly set up exactly right, don't you? And you look at those people as if they were insane. Ooh, what time is it? Good, I got time. Real quick story. Compartmentalization. So my wife, because she's not here so I can talk about her. My wife, we have a dishwasher. Do you guys have a dishwasher? Because thank God for dishwashers, right? You don't have a dishwasher. You are the dishwasher. Yeah, okay. That's why we have kids too. But you have a dishwasher. To me, I can load things into the dishwasher in any manner I want, and they will get washed. I know. My wife comes behind me and says, don't do that. The bowls belong here, the forks belong here, the knives belong here, the plates belong here. We've been living together for how long, and you still don't know this? I was like, but it's a dishwasher. It'll make the things clean. She said, no. There's a compartment for each thing. That's compartmentalization. Everything has its spot. Would it work? Non-compartmentalized? Yes, it would, because my wife's not here to tell me it won't. It would absolutely work. So that's prokaryote, non-compartmentalized, eukaryote, compartmentalized. I'll get through the structures of the prokaryotic cells. In prokaryotic cells, they all have 3 structures. Every single prokaryotic cell has 3 structures. They all have a plasma membrane. We've seen that before. What are those little circles with tails coming out of them? What? Yes, phospholipids. Those are those phospholipids we learned about. How would you describe a phospholipid? Yes, they're amphipathic. They both love and hate water. This phospholipid bilayer is responsible for sort of letting some things in and not others, for allowing proteins to embed in the surface for signaling. They will all also have a nucleoid region. This is not a nucleus. This is just a place where your DNA is held. It's sort of like your pile of sticks. This is where I keep my pile of DNA. Right there. It doesn't have a particular compartment to it. There's nothing binding it together. I'm just going to leave it there for now. I'm going to play with other stuff. The nucleoid region contains the cell's nucleic acids, the nucleic material, the genetic material. And they all have ribosomes. We are going to talk a lot about ribosomes as your first introduction. Ribosomes help make polypeptides. They take a strand of ribonucleic acid, RNA, and they're able to translate that into a strand of amino acids, polypeptide. What happens to the polypeptide after that? After you have a single long strand of amino acids? They interact with themselves, form secondary structures, and the secondary structures interact to form tertiary structures, and the tertiary structures interact quaternary structures. So all prokaryotes have ribosomes, a plasma membrane, and a nucleoid region. They may also have pili, flagella, a cell wall, or glycocalyx. Pili are used, this is sort of the optional structures. This is the deluxe version of the prokaryote. They might have these pili, and the pili are used for, one is for movement, the other is for sex. Because when you think tentacles, think bacteria sex. What happens is these little pili will reach out from one bacteria to another, and they touch each other. And when they touch, they send DNA back and forth. You might have flagella. Flagella are these whip-like structures off the end. They are also really good for movement. They help push the cell along. They spin around like a little motor, propelling the cell forward and propelling the cell backwards. They might have a cell wall, the strong layer on the outside of the cell in order to prevent osmotic lysing, or to allow these cells to survive in conditions they otherwise wouldn't be able to. Gives them protection, stops them from bursting like overfull balloons. And you might have glycocalyx. Glycocalyx is almost the slimy coating on the outside of bacteria that allows it to stick to things. Think, you guys brush your teeth, right? The reason you brush your teeth is 1, bad breath. 2, to get rid of the bacteria that are growing on your teeth. They're held to your teeth, those bacteria, by glycocalyx. It takes mechanical action of a toothbrush to remove it. Your tongue won't do it. And you know you've gone to school sometimes because you didn't have time to brush your teeth, and you're like, I'm gonna, get it off with your tongue. It won't work. You have to mechanically remove it because glycocalyx is so sticky, it will hold those bacteria to your teeth. It holds, you've seen creek beds with like algae growing on rocks, right? No matter how fast that water's moving, the green stuff's held there by glycocalyx. It's not going anywhere. So those are the optional structures in prokaryotes. So we're covering eukaryotic cells today and structures found in the eukaryotic cell. How many of you guys have seen a picture like this before? Yeah, most of you have. You remember bio in like 6th grade, 7th grade, 8th grade, and then high school? They were obsessed with, here's a picture, label it. That was all you guys had to do, and it was ridiculous. I'm never, ever, ever going to have you label a picture of a cell. I think it's silly because you guys have at this point looked through a microscope. You've seen cells. Do they look like that? No, of course not. That's a cartoon. It's a representation of it. So instead of having you guys label a cell, all I'm going to ask you to do is learn all of the organelles and how they relate to each other. And then discuss that in a big, long essay. I know, it's like, woo, more essays. Truth is, every single organelle, every little piece of this cell will communicate with every other piece. They all have a role. They all have something to do to keep the cell functioning. So all cells, all eukaryotic cells have these membrane-bound compartments called organelles. Organelle means little organ, right? The organelles are they have membranes surrounding them. What do you think those membranes are made of? Phospholipids. When in doubt, phospholipids. These amphipathic molecules automatically form bilayers, creating little tiny compartments where specialized things can happen. We're going to go through all the different components of the cell today. Starting on the outside edge of the plasma membrane, then working our way in from the nucleus, and then discovering the endomembrane system, how all of the proteins get moved through the cell, as well as how energy is produced within the cell. First part I want to talk about is that outer coating of the cell, the thing that separates the inside of the cell from the outside. It's called the plasma membrane. And again, you'll notice it's made up of a bunch of phospholipids. Remember I told you guys that stuff would keep coming into play. Come on. They're going to be not just phospholipids making up the plasma membrane, but also lots of different proteins. Lots of proteins that are anchored within it. These proteins all have hydrophilic regions and hydrophobic regions. Remember we talked about when proteins form, they've got some parts of them that are hydrophilic, that might be polar. Some parts that are hydrophobic, maybe a lot of hydrogen and carbon. The hydrophobic ones hate water. So what they do is, to avoid the water, they hide in the phospholipid bilayer. They hide in the tails. So by putting that hydrophobic region of the protein inside those tails, you're able to avoid water. And it's anchored there. It's not stuck there. It's not going anyplace. Those membranes that go all the way through are called transmembrane proteins. And some of them are going to act as channels. They'll let some things into the cell, but not others. That's called protein transport or plasma membrane transport. You have to get some materials from outside of the cell into the cell, and some things from inside of the cell to the outside of the cell. Because of the hydrophobic layer of the phospholipid bilayer, because of all those tails, you can't just go straight through it. You have to have a door. It's like you guys can't go right walking through that wall. No matter how much you want it, unless you're like the Kool-Aid man, then you can totally do it. But you can't walk through the wall. You need a door to get through. The proteins act as a door, allowing transport across the membrane. They let some things in, but not. These proteins may also be responsible for cell signaling. By being embedded on that plasma membrane, a signal from outside of the cell can interact with the exterior of the protein and release something on the inside of the cell that can then start a pathway, that can start things happening. Likewise, you can send a signal from inside of the cell out of the cell by releasing something. So you've got selective uptake, you've got cell signaling. The cell membrane is so important for adhesion, holding cells together. I'm not going to ask you about integrins or selectins or cadherins or the Ig superfamily. I put them up here, though, so you can take a look at them. What do these integrins look like? Hooks, right? Have you guys ever looked really closely at Velcro? Remember when you used to not know how to tie your shoes, or maybe you still don't know how to tie your shoes and you have Velcro? Velcro is made up of a whole bunch of little hooks, and those hooks will grab onto each other. Likewise, you've got these proteins that are embedded in the membrane with a hook on the end. You have another cell that has these embedded in with a hook on the end. So when they come together, they latch onto each other. Selectins act almost like a suction cup. The tip of it, this EGF-like domain, is going to latch onto a corresponding molecule on the other cell. Cadherins, I always think of cadherins acting almost like Batman's grappling hook. It'll pierce another cell, open up, so that it keeps the two cells together. And then the Ig superfamily has always worked like a tentacle to me. Those disulfide bridges are going to end up, this particular protein is going to wrap around and start grasping onto another cell and binding to it. I don't need you to know all of that. What I need you to notice, though, is they all have a certain form. And what does that form give them? Their function. In this case, the form of the hook gives it a function of adhering. You'll also note that all of them are coming into this cell membrane, this bilayer at the bottom. In order to get into the bilayer, you've got this hydrophilic region outside of the bilayer, a hydrophobic region on the inside of the bilayer, and then a hydrophilic region on the other side. There's water here, water up there, no water in the middle. That anchors it. It holds it steady and in place. Some cells are going to, in turn, have a cell wall. The cell wall is on the outside of the cell membrane. Not all eukaryotic cells have a cell wall. You saw a few in lab. What kind of cells have a cell wall that you noticed in lab? Plants. Plants have a cell wall. It's made up of all sorts of super strong carbohydrates that give it structure. So these are carbohydrates giving it structure. What do you expect the form of those carbohydrates to take if they're structural? What do you expect from all of the different the shapes that it would form? Let me put it a different way. We talked about 3 different types of carbohydrate macromolecules. Branched, unbranched, moderately branched. What kind is this going to be? Unbranched. There's no branches, because if it's not, there are no branches, water can't get in there to break it down. These are used for structure, so it's big, long, straight lines all stacked on each other. As we move in from the plasma, any questions on the plasma membrane before I go on? As we move in from the plasma membrane, we run into the cytosol. You've got the cytosol's the aqueous environment that takes up most of the cell's volume. And that excludes the nucleus and its organelles. The cytosol is filled with cytoplasm. It's that aqueous solution that fills the cytosol and some of the organelles. So the cytosol is the location, the cytoplasm is the gooey stuff, the aqueous solution. If it's an aqueous solution, what's it made of? Water. So we have our plasma membrane, and the plasma membrane encompasses the cytosol. Cytosol is important because this is where you're going to have the buildup or breaking down of macromolecules. What kind of reaction is going to be used to build a macromolecule? Yeah, condensation synthesis or dehydration reactions. And of course, to then break one, you're going to use a hydrolysis reaction. Most of those reactions are going to happen in the cytosol rather than at the membrane. You have two big kinds of reactions. Yes, hydrolysis and condensation, but you can broadly categorize them as either catabolism or anabolism. Catabolic reactions are reactions where you're breaking things down, you're catabolizing them. Typically, catabolic reactions release energy. So would condensation or condensation synthesis or hydrolysis be catabolism? Hydrolysis, because you're breaking things down. Once you break them down, you can release the stored energy to build other macromolecules, and you've recovered monomers to do the same. The reverse is anabolism. Anabolism builds macromolecules. Typically, this is going to require energy. You use monomers to build things. So if you have catabolism and anabolism and you put them together, you end up with metabolism. So when you hear about people talking about cellular metabolism, they're talking about all of the anabolic reactions and all of the catabolic reactions combined. All right, so now we've got this gooey aqueous solution in the middle, the cytoplasm. We got the plasma membrane surrounding it. But we need to give it shape. We need to give it structure. The cytoskeleton is responsible for providing the overall structure of the cell. It's going to be divided up into three components: microtubules, intermediate filaments, and actin filaments. Most people think of the cell as sort of this fluid-filled sac. It's malleable. And you guys have seen, or if not, you're going to see today, cells moving around. And they just look like they're balls that sort of squish and deform in all sorts of ways. There's actually a method to the madness. The reason those things are moving in all those different sorts of ways, the reasons they're deforming in those ways, is because different parts of their body are extending in different directions. It's just like with you. You have the skeleton on the inside of your body. That skeleton provides you a rigid shape. If you wanted to move your arm, you can. Your skeleton is helping with that. It's giving your arm shape. The first type of piece of the cytoskeleton that we want to talk about are the largest. And they're the largest even though they're called microtubules. These microtubules are big, long, hollow tubes. And ultimately, microtubules serve as a rail system within the cell. They help move things from point A to point B. They extend and they contract. They can push or pull things. They start forming at an organelle called the centrosome. The centrosome is an organelle whose whole purpose is serves as the site for microtubule formation. You may have heard of the centrosome described as two centrioles, these two little barrel-shaped structures at the bottom of a cell. And microtubules grow out of them. Animals have those. Plants don't have that. Plants have what's called a microtubule organizing region, which is shorthand for saying we don't know why it happens, but it happens. So you can grow microtubules out of centrosomes. The other component of the cytoskeleton are the intermediate filaments. These intermediate filaments provide rigidity and structure. So whereas the microtubules are a rail system to transport things through the cell, to move things around the cell, the intermediate filaments are what give it its rigid shape. They bind to each other, forming these rope-like structures. And by binding to each other and pushing against each other, they're able to provide a rigid structure on the outside of the cell. So if you think about it, here, Cassie, hold out your hands like this. So if I push on Cassie, there, good. She's holding all of my weight. Surprise. My back is rigid. You could push on my back, and because she's pushing this way, you can't squish us closer together unless you're super strong. Don't do that. Do you see what I'm saying? There's these two, the connection, those two rope-like structures are giving it support, and they're pushing on the outside edge. It's almost like, did you guys ever read when you were younger? I know it's sort of this thing that's going out of vogue. Like books. They don't have their own light or anything. All right. Did your parents ever tell you, it's time to go to bed, you can't read anymore? Some of you guys are like, no, never. My parents were just happy I could read. And this happened to me when I was younger. Did you guys ever make a tent in your room? How about that? You had to have done that. You made a tent fort, right? Tent fort. Did you ever make a tent fort at night? Did you ever take a flashlight in with you? Okay, if you got a tent fort, you got this. The poles of your tent fort, whether they were chairs or just your head, and you know you've made that tent fort recently probably, where it's just your head under the covers and everything's draping down, and you've got it like wrapped underneath you. Some of you guys are like, no. And some of you guys are like, yeah, that's Sundays. Your head or those chairs are those intermediate filaments. They are providing the supports. They're providing the strength and the rigidity. The rest of the tent can fall apart, but those supports, they're going to stay. Those are what are going to be the intermediate filaments. All of those anchor in the cell membrane. If they anchor in the cell membrane, so that means this section here, extending out, is hydrophilic, and over here, into the membrane, is hydrophobic. Provides a rigid anchor. And the last part of the cytoskeleton, the smallest parts, are the actin filaments. They are teeny tiny, and they sort of overlap each other all along the plasma membrane. The way I remember actin is I remember that actin causes actions. It's actin filaments that cause little bulges or deformations in the membrane of the cell. And I'll make it over here. So, oh, now I don't have chalk. There it is. So if I was to take this cell and extend something out, there's a whole bunch of actin filaments all along this edge overlapping. This part can start extending outward because the actin is growing there and shrinking down there, allowing for a bulge. Take a look at these slides, these stains here. The green stains in the right picture are going to be your intermediate filaments. The red stains are actin. So you can see the actin is localized to the exterior edge of the cell, whereas the intermediate filaments are all throughout. In order to move things from point A to point B in the cell, because everything is moving for a reason, you're going to need a motor protein. Whereas microtubules would be like the rail system, you need something to ride on the rails. You need some engine. You have these little proteins like kinesins or dynesins that literally walk along a protein. They have one shape. There's one shape's like this. Hold on, I can do this. I'm up on one foot. And then a phosphate binds. When that phosphate binds, it changes the shape of the protein. So now instead of being like this, it's like this. And then the phosphate releases, and when the phosphate releases, that goes down, that comes up, and then another phosphate binds and changes shape, and then that phosphate's released, and then it binds, and then it's released. And I'm not kidding, that's really what it looks like. Actually, it looks more like this circular motion, like this. And as these motor proteins are walking along, they're carrying behind them massive loads. They might be carrying vacuoles, or they might be carrying other molecules, macromolecules. But they move them from one point in the cell to another, following the microtubules. The cilia and flagella, these are going to be primary motive forces in the cell, are going to move because of motor proteins. So what happens is you've got these motor proteins that can grab on with one side and walk with the other. As they're on an intermediate filament, they're on I have to think about that. They're on an intermediate filament, and they're going to walk on one side. They're walking like this, and as they're walking, they're grabbing hold of the cell and pulling, and that causes a rotor action. So the end of it, the microtubule itself, begins to spin like this. The faster those motor proteins work, the faster it spins. The faster it spins, the more motion the cell gets. And this works for both cilia, which are these little tiny hair-like strands, and also for flagella, which are these big, long whips. The way I always think about it is, have you guys ever been in a boat with somebody who's a jerk, and they do that whole, like, rock the boat from side to side? Okay, a lot of you guys are like, yeah, and some of you guys are like, I totally do that. The idea is, if you're on a boat, here, this will be my boat. And I will definitely not die doing this. If you're on a boat, and you can pull one side, then the other, and eventually it builds up to the point where the whole boat is rocking back and forth with very, very minimal effort. These motor proteins are building on each other's force. So one motor protein doesn't do much, but as they're acting in concert, you get a big swaying back and forth. And as you have that swaying back and forth, there's that rotary action, and it's getting faster and faster and faster. If you've ever swung anything over your head in a circle, it's hard to start it swinging, but once it's going, you keep it going pretty easily. If you haven't, I don't know, swing a 2-liter bottle over your head sometime. Don't tell your friends what you're doing and why you're doing it. Just start swinging a 2-liter bottle. It's worth it. So that's all the components of the cytoskeleton. So we've got the plasma membrane. The plasma membrane is filled with the cytosol. And cytosol is made up of this cytoplasm, which is an aqueous solution. Within the plasma membrane, it's given structure and form by the cytoskeleton, made up of actin filaments, which cause actions along the exterior edge, intermediate filaments, which give it its shape, and microtubules, which act as a rail system on which motor proteins can walk. Now we're going to start talking about the endomembrane system, how we're going to have all of these organelles connected to each other. We've got the stage set. We've built the stage, the cell itself. Now, talk about each part. First part we're going to talk about is the nucleus. The nucleus is generally located as a central part of the cell. This is where you keep the cell's genetic material. You keep the DNA. What kind of macromolecule is DNA? It is a nucleic acid. So in this nucleus, you're going to protect, you're going to organize, you're going to replicate, and you're going to begin the expression of the genetic material. The genetic material is the plan to build you. Everything you need to build a human you is contained within the nucleus of any one of your cells. That's fantastic information. And you guys, I wish I could convey to you how awesome that is. Everything that makes you you is contained within each and every one of your cells. Some things are turned on, some things are turned off at different times. But it's because of the nucleus and its protective role and expressive role that you are you and not somebody else or not something else. The nucleus is surrounded by the nuclear envelope. The nuclear envelope is double membrane bound. It's not just a bilayer, it's a double bilayer. 2 bilayers. That means, and I made it. So you've got phospholipids and their tails, and then phospholipids and their tails. But wait, there's more, because that's 1 bilayer. It's a double bilayer. I am almost out of chalk. There we go. Double bilayer. Makes it super hard to get in and out of the nucleus. It is really well protected. The only way in and out is through these structures called nuclear pores. These pores are protein complexes that span the whole thing. So we've got water here, water here, water here. In order to get through without a nuclear pore, you'd have to go from a hydrophilic region to a hydrophobic to a hydrophilic to a hydrophobic to a hydrophilic region. It's extremely difficult. So instead, there are protein complexes that do that, and they go all through and make a tunnel. That tunnel is known as a nuclear pore. That's the way you get in and out of the nucleus. And it's highly regulated. So we've got this double membrane. Within the nucleus, the nucleic acids, the DNA, is highly condensed into a form called chromatin, or what you'll know it more as is chromosomes. Chromosomes are DNA that's wrapped around these proteins called histones and really, really condensed up. It's sort of like if I was to take off my shoe, you have a long shoelace. You can condense the shoelace. Here, in fact, make it here. There you go. You got long shoelace. You can condense it by just spinning it up. As you spin it, it gets thicker, and then it starts to get these new structures on it. And you'll see that it gets much smaller. So that's called supercoiling. You got a much tighter thing. Well, DNA does this not once, not twice, does this about 16 times. Coils and coils and coils and coils to get it so that it's supercoiled into becoming a chromosome. That's going to take a while to unwind. So the chromosomes live inside this. Inside the nucleus, you have a slightly different aqueous solution called the nuclear matrix. And you also have the nuclear lamina. Now, these nuclear laminas there as intermediate filaments. That's where you're going to hook your DNA. Your chromosomes aren't just floating around in there. They're literally attached to places. So you've got intermediate filaments acting again as a skeletal component, holding these chromosomes in place. Within the nucleus, you also have a nucleolus. This is a small region in the nucleus where you build ribosomes. So we have our nucleus. Nucleus is double membrane bound. It's got chromosomes in there. And it's got a nucleolus. What are ribosomes good for? Why do you need ribosomes? Based off of the last time we'd met. Yeah, they read that. Well, they help read the DNA. They're going to read another molecule called RNA. DNA gets transcribed to RNA. And then that RNA goes out to the ribosome where it's going to be translated into a polypeptide, into a protein. This is where you build your ribosomes. Those structures that all prokaryotes have, structures that all eukaryotes have. And like I said, those ribosomes are good for building proteins, for building polypeptides. I shouldn't say building proteins. They really just build polypeptides. What's the difference between a protein and a polypeptide? Other way. Protein's made of polypeptides. Oh, sorry, I just heard it wrong probably. How do you get a protein from a polypeptide? Following the hierarchy of structure. Primary structure being the polypeptide, which I'm pretty sure you said, Karen, and I just totally misinterpreted, so I'm sorry about that. Secondary structures where the primary structure interacts with itself. Tertiary structure where the secondary structures interact, like the alpha helices and the beta sheets. And then quaternary structures where two or more tertiary structures interact. In case you don't remember, I did tell you at the very, very beginning of the semester the entire final exam is all about proteins, right? Remember that? If not, surprise, now you get to remember. Everything on that final exam is the life and death of a protein, because everything we cover in this course is all about proteins. Ribosomes help to decode RNA to create a polypeptide. In order to get out of the nucleus, the only way a ribosome can leave is through the nuclear pore. So we're going to leave the nucleus and enter into this protein sorting pathway. All right, so now we've got our nucleus with our nucleolus and our, whatever these things are, and our chromosomes, sorry, with these exits, these nuclear pores. Again, the nucleus is double membrane bound, which is why it requires nuclear pores. From there, we proceed into the endomembrane system. Right next to the nucleus is the endoplasmic reticulum. This is a series of flattened membranes. It's a whole bunch of membranes. So if they're membranes, what are they made of? They're made of phospholipids. Inside of this, you've got this aqueous solution called the lumen. That's where it happens. You have two kinds of endoplasmic reticulum. Rough endoplasmic reticulum and smooth endoplasmic reticulum. What do you think the difference is? Yes. Yeah, you got ribosomes on one, and what does it make it look like? It makes it look rough. On the other one, you have no ribosomes, which makes it look smooth. The rough endoplasmic reticulum has all of these bumps all over it. Typically, the rough endoplasmic reticulum is right next to and almost contiguous with the nucleus. So if you were to draw the rough endoplasmic reticulum, sort of circles around the nucleus, like a bunch of arms like that. And on one part of it, there are a bunch of bumps. Those bumps are ribosomes. They're all attached to these tiny channels on the endoplasmic reticulum. So a ribosome will come and bind to the rough ER, and it will also bind to RNA, ribonucleic acid, produced from the nucleus. Both of those have left the nucleus to the nuclear pore. They meet up at the endoplasmic reticulum. When they meet, this green structure down here, that's the RNA. The purple structure is the ribosome. The RNA begins to be read and translated into a polypeptide that's fed into the endoplasmic reticulum. So now we have polypeptides in the endoplasmic reticulum. All right, this is our first step in building a protein, right? We got our polypeptide. That rough endoplasmic reticulum serves three big functions. Protein sorting. It sorts out proteins that are destined for other organelles. Not everything's going to end its life in the endoplasmic reticulum. It inserts newly made proteins into the endoplasmic reticulum. That means that some proteins get embedded in the membrane. We're going to talk about how that happens in the next lecture, but it's super cool, trust me on that. And it performs glycosylation. Glycosylation is one of my favorite words in the world. I love the way it just rolls off the tongue and says glycosylation. So glyco, when you think glyco, what do you think? Where have you heard that before? What? Glycocalyx. Same idea. Glyco. Think glycogen, glucose, sugars. Carbohydrates is what it is. All glycosylation means is adding a carbohydrate. Adding a whole bunch of carbon and hydrogen and oxygen to something. So you can stick that onto a protein or a lipid. The smooth endoplasmic reticulum, on the other hand, increases surface area for the enzymes to metabolize products. Now, this is a common theme that you're going to see in biology. Whenever you see twists and turns, just across the board, when an organism has twists and turns, it's to increase surface area. Have you ever seen an intestine before? I don't know why you would, but you might have watched a show that has intestines in it. Okay, surprisingly enough, a lot of you are nodding, so that's good. If you've seen intestines, I don't know, somebody pulling out their own intestines, you'll notice that they're really, really super long. It's because they can twist and turn. Your intestines are about 30 feet long. You're not 30 feet long, but they go back and forth and back and forth and back and forth inside of you. That's to give your food longer time to digest. Much easier than it going in your mouth and straight out the other side. Gives it some time. You know it takes anywhere from 12 to 36 hours for you to digest something. So when you go home, you eat some dinner and you're like, man, I have to have a bowel movement. What's coming out is not what you just put in. I know you know that, but there are a lot of people who are like, no, I just ate corn and I know it's coming out right now. That's not the way it works. 12 to 36 hours because of all those twists and turns. The smooth endoplasmic reticulum has all of these twists and turns back and forth because wherever there are those twists and turns, the longer the protein stays in there, the more reactions that can occur on it. Lots of things can happen. This is where you can create phospholipids. This is where phospholipids get built. You can add functional groups to your polypeptide. Any metabolic reaction that you need to happen can happen in the smooth endoplasmic reticulum. Once you're at the smooth endoplasmic reticulum, you've gotten to sort of the end of that particular organelle. You're going to the Golgi apparatus next. Again, another series of discs. But the problem is they're separated by distance. You got to get from one to the other. The Golgi apparatus is sort of the, final sorting place. Think of it like, you guys have ordered things from Amazon before, I assume, right? Because why wouldn't you? You order something on Amazon. The order goes into this large central headquarters. From there, that large central headquarters, which is processing your order, is like the endoplasmic reticulum. But then it doesn't go straight to your house from there. Then it goes to a distribution center. So from there, usually that's the one where you see it stay for a few days as you eagerly await it arriving. That distribution center would be the Golgi apparatus. So here is where it's originally found in the endoplasmic reticulum. Then it gets shipped over to the distribution center, the Golgi apparatus, and then from there it gets shipped to its final destination. In the Golgi apparatus, you get two big things happening. More glycosylation, because you can never get enough glycosylation. Add more carbohydrates on. The other thing that can happen is proteolysis. All right, proteo, what does it look like? Word. Protein. Lysis means what? Break apart. So we've got proteolysis, breaking apart proteins. You're taking a polypeptide in the Golgi apparatus and splitting it. I know it sounds weird to do. Why would you build it up just to break it later? And to answer that question, the big question of why would you do that, I turn your attention to baking cookies. Have you guys ever baked cookies? Some of you have. And there's a reason you bake cookies. It's because you freaking want cookies. Or you've been guilted into like a bake sale or something. But you don't want an entire batch of cookies, right? You just want one or two cookies. Okay, some of you guys are like, yeah, no, I want the whole freaking batch of cookies. That's why I'm making cookies. But you may not want the whole batch of cookies. You just want like one or two cookies. But you don't just make one or two cookies. Just make the giant ones. You make a whole batch. Why do you make a whole batch instead of just two cookies? If you want some later, that's one big reason. The other reason is it's just as easy to make 20 cookies as it is to make 2. The difficulty level is the same. You're using about the same amount of stuff. You're not going to, the cookie mix, whatever you may use, isn't something you can necessarily split down. So instead of just making two individual really, really, really small batches of cookies, you're going to make a whole batch. Because it's just as easy to make a whole batch as it is to make one. And it leaves you leftovers. And leftovers are the greatest thing in the world. So proteolysis is where you're going to cut up peptides to make smaller peptides. The Golgi apparatus is also important for secretion, for getting stuff out of the cell. You want to send signals, you're going to send them from the Golgi. There's going to be pieces of that Golgi apparatus that bud off. what's going to cause that budding? What's going to cause that change in the membrane around the Golgi? Let me draw a picture. So if I have a Golgi apparatus, so here's our arm of the endoplasmic reticulum, here's our Golgi right there. And I want to move something from here to here, or from there to there. I've got a protein right there. I'm going to start to bud this off. I'm changing the shape of that membrane. What am I using to change the shape of that membrane? What's causing that change in shape from earlier in today's lecture? Say it again. All right, here's what's causing that action to occur? Actin filaments. Actin filaments are, anytime you see a change in the plasma membrane, think actin filaments. A ring of actin is forming around it, getting it tighter and tighter and tighter. Think of a belt around a very, very large person. You keep squeezing that belt, you keep pulling and pulling and pulling, eventually you're going to cut that person in half. Actin is forming a belt and getting tighter and tighter. And as it's getting tighter and tighter, that pops off and forms what's known as a vesicle. That vesicle contains inside of it a protein, something. It then moves over to the Golgi apparatus. What's it moving along? It's using a motor protein to get from point A to point B, but what's that motor protein walking on to get you there? It's got a microtubule, the rail system. So then it gets over here. It binds to the Golgi and allows that protein on the inside. If you wanted to go from the Golgi to the outside with secretion, here's the outside of the cell. Going to do the same thing. You bud off. You have this ring of actin. It pops it off. It moves over here. It fuses with the membrane, and it pushes that protein out, moving it to the extracellular environment. So the Golgi is sort of that final distribution center. Sends things out. So the cells are going to be further modified later on. I'm sorry, the proteins will be further modified later on. They're moved from point A to point B. And they can go to all sorts of organelles in the cell. So the three big things the Golgi apparatus does: glycosylation, protein sorting, and secretion. From there, it all matters on what your protein is, what it's going to do, what its function is. It can go to all sorts of other organelles. It might go to a lysosome. A lysosome is important for cutting, for recycling macromolecules, for cutting them. The word lyse, where have we seen it before? Or lysis, how about that? Hydrolysis, right? We've seen it before, it's good. It means to break apart. Here, we're going to break things apart, the recycling center of the cell. You want to get these macromolecules broken down. Lysosomes have inside them a very specialized environment. That's one reason you have these organelles, to create specialized environments. They have enzymes in them called acid hydrolases. They were put here by that secretory pathway we just talked about, from the Golgi apparatus to the lysosome. Acid hydrolases are going to reduce the amount of energy you need for a hydrolysis reaction to occur. They make it so hydrolysis speeds up, goes faster. It operates most efficiently at about pH of 4.8. So you have these enzymes that work really, really, really well to cut things apart at pH of 4.8. Is pH 4.8 acidic or basic? It's acidic. How does it keep the pH at 4.8? With buffers. Well done. Questions so far? All right. Here's a big question. We have these very, very acidic, highly acidic lysosomes inside of our cells. What would happen if the lysosome was to burst and break inside of the cell? You throw off your pH. pH balance. You've been watching deodorant commercials. I hear about pH balancing in deodorants. I'm just like, I've done a lot of chemistry. I have no idea what that means. So, your pH is thrown off. So what happens to the cell? So its function begins to deteriorate, except it doesn't. Why doesn't the cell's function break down? Why doesn't the cell die from all of these acids released into it? Because when in doubt, buffers. Because buffers maintain the cell's internal pH. That's why buffers are so important. So we got these recycling centers going around, breaking down macromolecules that are no longer useful. We have another type of organelle called a vacuole. Vacuoles are areas of storage. Now they got actually three big purposes. You can store things in them, you can regulate cell volume, or you can help digest things, like little tiny stomachs. First idea is material storage. Plants have a gigantic vacuole. Part of the reason they do is because they're always making food with their chloroplasts, glucose. That glucose is being churned out, and it has to be stored somewhere, and it'll be used later. Animals like us, we don't have need for storage. We kind of live in the moment. So we have tiny, tiny, tiny storage vacuoles. Keep whatever we need on hand at that point, and if we need more, what do? What happens when you guys need more energy? You go eat something else. Isn't it cool being top of the food chain? Where it's like, man, I need more energy. Hum, 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 hum. Okay, I've got it. Plants don't get that option. It's good for regulating the cell's volume. We're going to learn in the next lecture, or the next series of lectures, about osmosis and diffusion. Water is going to constantly be flooding into cells. In order to stop that cell from bursting like an overripe balloon, I don't know if balloons get ripe, bursting like a balloon when you fill it with too much water, you have to push it out using an active contractile vacuole. So this vacuole right in the first one, it's filled with water, you use some energy, and you squeeze it out. Otherwise, water would keep flooding in. You guys have made water balloons before, right? There are three kinds of water balloons. There's the kind where you start filling it up, and you don't leave it on there long enough, and you tie it off. What happens to those? They're filled with a little bit of water, not much. You throw them at somebody, what happens? It bounces off, you had a perfect shot, and you messed it up because of your balloon. The other kinds are the ones that are just right. They're you fill them up moderately, you throw it at somebody, it blows up on them, but it doesn't get you wet. Those are the best kind. Third kind are the ones that you screw up right at the beginning. You overfill them with water. You put it onto the sink or the hose or whatever you're trying to fill up the balloon with, and the balloon gets huge and then pops all over you. The contractile vacuole prevents cells from filling up with water and then popping. Otherwise, they would. And you could see it happen. When nurses make mistakes, let's say they there's somebody who's dehydrated, and this is like for some reason people make this mistake a lot. They're like, they're dehydrated, quick, give them intravenous water. It's a really bad idea, because what will happen is all of that pure water around their blood cells will flood into their blood cells. Yes, they will be rehydrated instantly. The problem is their blood cells will just start popping like balloons. Very painful. And finally, it can assist with digestion. Phagocytosis is it's a fancy word for digestion in cells. What happens is the cell membrane begins to deform. Okay. I'll just draw it. And it makes a little bulge. Let's say there's a food particle right here. As it's going into the cell, the membrane begins to form around that food particle until it eventually pops off on the inside. Now you have a vacuole that's a digestive vacuole. What caused the plasma membranes to deform like that, to have that shape? What causes changes in the shape of the plasma membrane? actin. Actin filaments causes changes in shape. Actin causes actions. So vacuoles are good for digestion with phagocytosis. They're good for storage, and they're good for maintaining cell pressure. Another type of vacuole is the peroxisome. Peroxisome, what does it sound like? Peroxide, like hydrogen peroxide, right? What the peroxisome does is it breaks down molecules by either adding oxygen or removing hydrogen. This is what gives you your ability to stop hydrogen peroxide from damaging your cells. We talked briefly before about hydrogen peroxide as a free radical. The peroxisome breaks down hydrogen peroxide. So even if you pour hydrogen peroxide on your arm, it'll kill the bacteria, but it won't kill you, because this thing is churning away these proteins that are called catalase. They take H2O2, hydrogen peroxide, and convert it to water and oxygen. Without that catalase, you break down H2O2 into a free radical, and then you get damage. Two more organelles, then we're done. The chloroplast and the mitochondria. All right. The mitochondria. Now, I want to point this out because this is I have a few pet peeves. One pet peeve is this whole brown eye, blue eyes thing in genetics. I really hate that. The other pet peeve I have is this idea that the mitochondria is the powerhouse of the cell. If you were to write the mitochondria as the powerhouse of the cell on your exam, I will just mark it wrong. That's how much I hate this idea. Because each of you has heard sometime in your life that the mitochondria is the powerhouse of the cell, haven't you? And they're like, remember, the mitochondria is the powerhouse of the cell. What does that mean? It's not like it's making electricity. It's not like it's doing much anyway. What mitochondria is the site of aerobic cellular respiration. Write that down, learn that. That's how I want you to describe mitochondria. The site of aerobic cellular respiration. This is where we're going to build the majority of our adenosine triphosphate, ATP. First time we're seeing that word, but it sure is not the last. It's going to come up again and again and again and again for the next several 2 months. 2 months. Yep, just 2 months. It's the site of aerobic. So think aerobics, A-E-R-O-B-I-C. Cellular, C-L-L-U-A-R. Oh, there was an L at the end of that, sorry. Respiration. Now, the cool thing about mitochondria, and this is that there are hundreds to thousands of them in any given cell. And because of that, that's the big reason I don't have you guys drawing pictures of the cell or labeling pictures of the cell. Because I swear to you, I felt so cheated when I got to graduate school and I actually stained mitochondria. Here, see if I got this right. A is, I want to say, it's intestinal lining tissue. B is cheek cells, so the cells on the inside of your cheek. C is heart cells. What they've stained are the individual mitochondria. Now, each tiny green dot is a single mitochondria, or mitochondrion, I guess. There are so many of them here, you can't see anything else. Thousands upon thousands upon thousands. And they are teeny, teeny, tiny. Which is why, typically when you draw a picture of the cell, you've got all of these things, and the mitochondria is like the size of my fist. It's nowhere near that size. I felt so gypped. I felt like I was lied to. So I want to be up front with you guys. The mitochondria are really small, and there are hundreds or thousands of them in any given cell. It all depends on how much energy that cell needs. Your cheek cells don't need much energy. Your heart cells need a lot. Now, the mitochondria is a lot like the nucleus. It's got double membrane. Other cool thing is Yeah, it's also got lots and lots of twists and turns in it. Why does it have twists and turns? What does that do? Increases the surface area. And if you've increased the surface area, you allow more reactions to happen. So it's this double membrane-bound organelle. Wherever you've got twists and turns, you can have aerobic cellular respiration happening to give you lots and lots of ATP, these energy-containing molecules. And the final organelle is the chloroplast. This is the site of photosynthesis, where you're taking light and converting it into usable cellular energy in bonds. Specifically, you're making large organic molecules like sugar, glucose. Chloroplasts are green. That's a good way of noting them. They're fairly large. They, too, have both an inner and an outer membrane, just like mitochondria. And they, too, have lots of twists and turns. And again, why do they have lots of twists and turns? More surface area, more reactions, more sugar. The chloroplast and the mitochondria are linked to each other. The chloroplast produces sugar. The mitochondria takes that sugar and breaks it down. Does that make sense? Animal cells and plant cells all have mitochondria. It's only the plant cells and a few few eukaryotic cells, other cells that have chloroplasts in them. Chloroplasts and mitochondria will each have their own unique sets of DNA inside of them, different than the DNA in the nucleus. And they're going to independently reproduce. And they don't reproduce when the rest of the cell reproduces. They reproduce based on the needs of the cell. So if you think about, you guys ever had like a plant under a tarp over the winter? When you take that tarp away, what color is the plant? If it's dead, it's brown, I guess. I'm thinking more white. Have you guys ever seen like you let a plant grow for too long in the dark and it turns white? It doesn't have chloroplasts because the sun's not shining on it. It doesn't need them. But suddenly, once the sun starts to shine on it, what happens to that plant? It starts to turn green because the chloroplasts begin to very independently and quickly reproduce themselves. It's like you guys, when you exercise, or if you've thought about exercising, I'm sure. You probably have thought about it. You thought to yourself, man, I should really go out and exercise. If you've ever exercised at some point in your life, you realize that the first time you go out to exercise, you don't have much energy. You're like, I can't do this. The next time you go, it gets a little easier. The time after that, it gets even easier. Because as you stress your cells, as you make your cells use energy, the mitochondria say, oh, I should be working. And they double. They start to reproduce themselves in order to produce more energy. So by using energy, your body decides, hey, I need more energy, and it produces more of these mitochondria to make more energy happen. This doesn't need to happen during the reproduction of the cell. It happens outside of the cell's reproductive cycle. It's as if the mitochondria and the chloroplast were little tiny organisms living inside of you within the cell, which is kind of how we think they happened. This leads to this idea of endosymbiosis theory. I promise you, endosymbiosis theory will come up not just this semester, but next semester as well if you take 102. The idea of endosymbiosis is that a long, long time ago, you had eukaryotic cells, single membrane on the outside. A eukaryotic cell ate a bacteria. It's probably a brown algae. And that bacteria happened to be really, really good at producing energy. So it swallowed it up, formed a vacuole around it. So now you have this bacteria with its membrane surrounded by another membrane. So it's a double membrane. Rather than digesting that bacteria, the cell kept it alive, and it became sort of this little battery on the inside of the cell, churning out energy. So endo means inside, symbiosis means living together. So you have a bacteria living inside of this organism. The organism gets energy, the bacteria gets protection and food. Later, another another algae comes along. This one's a green algae. It gets eaten by the cell. Well, this green algae is super good at producing sugar. Instead of digesting that algae, the algae lives inside the cell, producing sugar for the cell, which is then broken down by the brown algae on the other side of the cell, producing energy. So this cell has it made. It doesn't have to do anything. It's just sort of keeping these two other things alive that are doing all the work. That's the endosymbiosis theory. That's how we think at this point that we had chloroplast and mitochondria in our eukaryotic cells. All right, before you pack up, and I knew you want to, I want to go through this whole thing one more time, because you know you have a big question on this. There's the chalk. All right, so we start off on the outside of the cell. What is the structure on the outside of the cell? What is it? The cell wall? Okay. In certain things, what else is there? Plasma membrane, right? So we got the plasma membrane. What's the plasma membrane made of? Phospholipids. And what's its role? Let some things in, let some things out, protect the cell, cell signaling, cell adhesion. Inside of the cell, you have cytosol and cytosol that's partially composed of cytoplasm, that aqueous solution. That's where you're going to have catabolic reactions and anabolic reactions occurring for the most part. The center of the cell has a double membrane-bound organelle. What's that called? The nucleus. The nucleus contains within it the DNA, the nucleic acids. In what form? How are they stored? What? Chromosomes. Chromatin, actually, you're exactly right. Chromatin, only chromosomes at some point in their life cycle. So you've got these condensed DNA. What else is in there? Something else. Little ball. Nucleolus. And that nucleolus is going to make ribosomes. So you have DNA that can produce RNA and ribosomes being produced by the nucleolus. Now they need to get out of the nucleus. How do they get out of the nucleus? Through the nuclear pores that go the whole way through. Those are going to meet up at this next structure that surrounds the nucleus. And what's that structure called? That completely surrounds the nucleus. The endoplasmic reticulum. The ribosome will come out and bind to it. The RNA will bind to that. Now you got these bumps on it. Because there are bumps, what's it called? The rough endoplasmic reticulum. This will allow, when the RNA and the ribosome meet, they're going to thread a polypeptide into the endoplasmic reticulum. So now you've got some protein sorting going on. You might have inserted it into the membrane itself. That protein will then move, or that polypeptide will move from the rough endoplasmic reticulum to the smooth endoplasmic reticulum, where there are lots and lots of twists and turns. What are those twists and turns for? To increase surface area. That allows functional groups to be added. You might make some phospholipids here. From there, you're going to take this polypeptide and move it over to the Golgi apparatus. That's going to be a centralized distribution point. Proteolysis happens there, and glycosylation happens there. Glycosylation being the attachment of a carbohydrate. Proteolysis is breaking that polypeptide into two. To get from here to here, you need to form a vesicle. So it's going to pop off. What's forming that vesicle? How do you get it to break off like that? Changes in the membrane. Actin filaments cause changes in that membrane, causing it to pop off. Well, now we have to get it from here to there. How's it going to get there? What's it have to ride on? Microtubules. And what's going to carry it on those microtubules? The engine that gets it to move. Motor proteins. Good. So you got these motor proteins pulling that vesicle from one point to another. They merge together, and you've dropped off your polypeptide in there. Glycosylation happens, protein sorting happens, proteolysis happens. Then you get shipped all over the place. You might go over here to a peroxisome. Peroxisome is going to be good for, you might be at that point a catalase protein, say. You might be able to break down hydrogen peroxide by adding oxygen or removing hydrogen. You might go to a lysosome. What's a lysosome good for? Lyso. Lyse means, yeah, hydrolysis happens there. You cut things up, you break them down. It's the recycling center of the cell. So the lysosome is generally pretty acidic. What maintains its acidity? Buffers. And if it was to burst and rupture and flood through the cell, what would happen to the cell? What? It'd be fine. Why? Because of buffers. So that's really good for recycling macromolecules, to break them down to individual monomers so they can be reused. Might go to a vacuole. Vacuoles are good for storage. They're good for digestion, helping in digestion. They're also good for maintaining cell volume. They might end up at a chloroplast, let's say a mitochondria, which should not be referred to as the powerhouse of the cell, but rather it's called what? Site of aerobic cellular respiration. Now it's double membrane bound, which gives all these twists and turns, which is good for what? More surface area, so you can have more reactions. You might have another double membraned organelle called the chloroplast. What's chloroplast good for? Making sugar. The sugar from the chloroplast is going to be used at the mitochondria in order to break it down to give you adenosine triphosphate, so you got some energy to do stuff. Something I didn't mention, but there's another organelle, the centrosome. What's the centrosome good for? It was mentioned on one slide some point way earlier. Here, I'll give you a hint. So it's going to be made up of these two centrioles on either side of the cell. That's where you're going to form microtubules, microtubule organizing regions. Okay, so there you go. The lecture in a nutshell. You're going to have to describe how that works. I guarantee you on the exam you're going to be asked about each and every one of these organelles and what they do. And I know your big question this time is to explain what I just said, which I assume is going to probably be a lot easier this semester because I just recorded it. But do you guys have questions? Your next big question will be done a week from today.